fermentation. So fermentation is also a type of process where some amount of ATP can be generated. Fermentation is again divided in two categories. First, we have lactic acid fermentation and alcohol fermentation. Now, unlike aerobic and anaerobic cellular respiration, fermentation actually doesn't involve respiration of any kind. So we don't observe an electron transport chain in fermentation, while this is observed in both aerobic and anaerobic cellular respiration. In both the forms of fermentation, glycolysis is the first step. So breakdown of glucose to produce pyruvate is the initial step in both the forms of fermentation. Now glycolysis is an ancient pathway and is observed in all the organisms. So depending on the fate of pyruvate, we have two different types of fermentation, which could be lactic acid or alcohol fermentation. Let's start with alcohol fermentation. Alcohol fermentation is commonly observed in the brewer's yeast. Brewer's yeast is a common name for the organism Saccharomyces cerevisiae. So Saccharomyces cerevisiae is an important unicellular yeast that belongs to the kingdom fungi. It is a unicellular organism and it can undergo alcohol fermentation. The first step here is again the conversion of glucose to two molecules of pyruvate. So we have two molecules of pyruvate, which is a 3-carbon compound, CH3. This is the structure of pyruvate. Now pyruvate undergoes a decarboxylation reaction. Decarboxylation because it is losing a molecule of carbon dioxide. Decarboxylation to produce two molecules of acetaldehyde. CH3CHO. So acetaldehyde contains a functional group that is aldehyde and its molecular formula is CH3CHO. Now acetaldehyde is the electron acceptor as it accepts electrons from NADH and gets converted into ethanol. Which is C2H5OH. And in this process, NADH gets converted back into its oxidized form, which is NAD+. So pyruvate, which is a 3-carbon molecule and produced through glycolysis, undergoes a decarboxylation reaction producing acetaldehyde, which is an aldehyde. Acetaldehyde can accept electrons from NADH, thereby producing ethanol or ethyl alcohol. And in this process, it is able to regenerate NAD+. So glycolysis requires the presence of NAD+ to continue. This reaction is especially important in the brewing industry as this helps to produce ethanol. The same yeast is also extensively used in the baking industry because the carbon dioxide which is released in this process is what actually causes the bread to rise during baking. Alcohol fermentation has two big advantages to the industry. One is through the generation of ethanol, which is routinely employed in the brewing industry as in the preparation of beers and wine. Second is in the baking industry where carbon dioxide helps the bread to rise. The other form of fermentation is called lactic acid fermentation. The muscle cells in the human body and bacteria, namely lactic acid bacteria, which is also known as LAB, stands for lactic acid bacteria, predominantly use this mode of fermentation to produce lactate. Similar to alcohol fermentation, this also takes place in 2 steps, starting with glycolysis. So in glycolysis, glucose gets broken down into pyruvate. So let's start with pyruvate. Here, pyruvate itself accepts the electrons from NADH and gets converted into lactate. C3H6O3. Again, NADH loses its electrons to pyruvate, thereby regenerating NAD plus back. Okay, so we don't have a decarboxylation step here. The pyruvate produced through glycolysis itself acts as an electron acceptor, accepting the high energy electrons from NADH and gets reduced into lactate. Now this step is again crucial as it allows NAD plus to be regenerated, thereby allowing glycolysis to continue and produce some molecules of ATP. So both the forms of fermentation are actually helping to regenerate this important electron carrier, which is NAD+. Whenever we are doing a vigorous physical activity like running a marathon or even a short sprint, the muscle cells will also carry out this mode of fermentation as oxygen becomes limiting in these conditions. The body is not able to get as much oxygen as it requires, and in these conditions the body or the muscle cells will also temporarily switch to producing lactic acid. So some amount of ATP will be released here, and that will be utilized by the muscles to carry out its further work. Now what do you think will happen to the lactate that actually builds up in the muscles during this activity? Pause for a moment and think what could be the fate of lactate produced in the muscle cells. The lactate actually gets converted back into pyruvate. So pyruvate is a 3-carbon compound, and this pyruvate can again be fed into aerobic cellular respiration. So production of lactate is just a short-term process, and whatever lactate gets produced travels to the liver where it gets converted back into pyruvate. Now we know pyruvate can then enter into the next phase of cellular respiration, which is Krebs cycle. So to conclude, we have aerobic respiration, we have anaerobic respiration, and we also have fermentation. In aerobic and anaerobic cellular respiration, it starts with glycolysis. Then we have the Krebs cycle or TCA cycle and the electron transport chain. So all these events are common to both aerobic and anaerobic respiration. The only difference being oxygen acts as the terminal electron acceptor here, whereas sulfur or sulfate can act as the terminal electron acceptor for anaerobic cellular respiration. Now fermentation is different because it only involves glycolysis from the above. Only glycolysis takes place in fermentation, plus the additional steps that take place is only to regenerate NAD+, so that it allows glycolysis to further operate. If you found this video helpful, please do share it with your friends.